Welcome to Lab Talk, a special edition podcast produced by the Scientist Creative Services team, where we explore topics at the leading edge of innovative research. This episode is brought to you by Isoplexis, a life science technology company that accelerates the development of curative medicines and precision therapeutics. Their award-winning single-cell proteomic systems reveal unique biological activity in small subsets of cells, allowing researchers to connect more directly to in vivo biology and develop more precise therapies. Their integrated systems were named Top in Innovation or Design by The Scientist, Fierce, Big Innovation, Red Dot, and multiple other companies. These systems are used globally to advance the field of single-cell biology as their customers overcome the challenges of complex diseases. Their products have been adopted by researchers around the world, including the top 15 global pharmaceutical companies and 45% of comprehensive cancer centers. In this episode, Niki Spahich from the Scientist Creative Services team spoke with Katie McKenna, postdoctoral fellow in the laboratory of Malcolm Brenner at the Center for Cell and Gene Therapy at Baylor College of Medicine, about her work developing a combination CAR T-cell immunotherapy and oncolytic virotherapy to treat solid tumors.
Chimeric antigen receptor T-cell immunotherapies hold great promise for cancer treatment. To develop these therapies, researchers engineered T-cells that express chimeric antigen receptors, or CARs, that recognize specific factors, or antigens, on the surface of cancer cells. Once bound to an antigen, the altered T-cells proliferate and induce a cytotoxic response to kill the target. Scientists can develop this immunotherapy from a person's own T-cells and reinfuse them back into the patient. Researchers hope to develop CAR T-cell therapies for a range of cancers by designing CAR constructs that target the unique antigens expressed on cancer cell surfaces. So far, CAR T therapy is approved to treat several forms of lymphoma, but the success rate in blood cancers has not translated to solid tumors. The cells that make up one solid tumor do not express a universal surface antigen, so the CAR T-cells have trouble identifying their intended target. T-cells also have trouble trafficking into and persisting among solid tumors.
Much of these problems are attributed to the tumor microenvironment, or that TME. It's a pretty heterogeneous network, not only malignant tumor cells, but also stromal cells that support this tumor, or also extracellular matrix, as well as immune cells and other inhibitory factors that can be produced by the tumor, or immunosuppressive subset of cells there that are inhibiting some type of effector cells that you want, like our CAR T cells. So this tumor microenvironment provides a lot of obstacles in order to have a very efficacious therapy. What we need to do is skew our research to focusing on how can we target not only our malignant cells, but also different components of the tumor microenvironment.
McKenna recently tested a cancer treatment using CAR T cells that target HER2, a receptor widely expressed in many tumor types, including breast, lung, and sarcoma. This CAR shows promising results in clinical trials conducted at Baylor for sarcoma treatment. McKenna wanted to increase the efficacy of the HER2 targeting CAR T cells in solid tumors, so she added viruses that further stimulate the immune response to the existing treatment and tested the combination in vitro and in vivo. Her work builds off of two previous studies from her colleagues at Baylor, Masataka Suzuki and Valentina Hoyos. Suzuki developed two adenoviruses, relatives of the viruses that cause the common cold, that together eliminate cancer cells. One adenovirus was oncolytic, or directly responsible for killing cancer cells, while the other helper virus expressed genes that stimulated the immune response. The helper adenovirus encodes two immunostimulatory components, IL-12, which enhances the T-cell response, and anti-PD-L1, which prevents inhibition of the immune system. Suzuki found that these two viruses, collectively referred to as CAD, can infect both tumor and stromal cells and boost the activity of the HER2 targeting CAR T cells.
They tested this system in a head and neck cancer to where they could actually deliver the CAD virus directly to the tumor. So they did intratumor injection, and then they followed it with the HER2 CAR T cell, and they were able to see really beautiful results where they could eliminate not only the primary tumor, but they could also eliminate metastases by allowing the CAR T cells to target any types of metastatic sites. That was super encouraging in terms that they could see this benefit of the combination therapy. What we wanted to do is develop our system to allow for targeting different tumors. That led to systemic administration of our virus, followed by the CAR T cells. The downfall with this is that many of us in the public have actually been exposed to adenovirus in terms of generating a respiratory illness, like a cold. So because of that, a lot of us have pre-existing anti-adenoviral immunity. And when you deliver a naked virus systemically, you can activate your pre-existing immunity or neutralizing antibodies that would then prevent the virus ever reaching the tumor.
To shield the viruses from immune recognition and deliver them to the appropriate site, McKenna pursued a Trojan horse strategy using mesenchymal stromal cells, or MSCs. MSCs home in on sites of inflammation, such as areas surrounding tumors, and they provide an environment for viral replication. Hoyos previously found that MSCs could replicate a different oncolytic and helper adenovirus system and release the viruses directly at tumor sites to avoid neutralization by the immune system. McKenna packaged Suzuki's CAD system inside MSCs. Once delivered, she found that the viruses spread throughout the tumor and its microenvironment, locally releasing IL-12 and anti-PD-L1.
We then infuse CAR T cells that can respond to the immunostimulatory components as well as target tumor. In this particular study, we are targeting lung cancer. When you infuse cells systemically, intravenously, one of the first sites where these cells go are actually to the lung. So we're utilizing that advantage in our initial studies that the MSCs do home to that tumor site, release the virus there, and then allows for CAR T cells to traffic to that tumor site. Our previous studies have shown that MSCs that do deliver our oncolytic adenoviral system only release the virus within the tumor site, and we don't detect it in other tissues.
Researchers typically first test cancer therapies in 2D cell culture. While this is a well-established starting point to determine if a treatment has potential, solid tumors are 3D structures, so conventional tissue culture leaves a lot to be desired. To complement 2D experiments and assess how the viruses spread throughout a solid mass, McKenna also grew 3D tumor spheroids from non-small cell lung cancer cells to mimic a solid tumor in vitro. In a third set of experiments, she tested the combination adenoviral and CAR T cell therapy in a mouse model of lung cancer. McKenna designed these experiments to mimic real clinical cancer therapies, allowing the proper time for MSCs to deliver the adenoviruses to cells or to tumors within the mouse, and for the viruses to lyse from those MSCs, infect tumor cells, replicate, and release the immunostimulatory factors. Once those steps were accomplished, she administered the HER2 CAR T cells and assessed their ability to kill cancer cells and shrink tumors.
We could look at reduced tumor cells in vitro. A lot of our assays used flow cytometry to do that. In our spheroid models, we utilized live image analysis to visually see destruction of our spheroid, and also used a marker for apoptosis, and we saw increased staining of that over time. And then in our in vivo models, we were able to track our tumor signal through a luciferase tag, and we were able to look at that enzyme signal through bioluminescence imaging. And so in each of these cases, we saw a reduction in our residual tumor over time, and it was further enhanced when we combined our MSCs carrying oncolytic virus in combination with our HER2 specific CAR. The initial assays showed us that we really were enhancing the anti-tumor effect of our CAR T cells. That's when we tried to really elucidate what is the mechanism improving our CAR T cells. That's when I utilized some functional proteomic assays by utilizing the isoplexis system, where we're able to measure secretion of many different cytokines from single cells. I was able to then isolate the T cells after 24 hours post T cell addition, and then I looked at their functional cytokine secretion, either with the CAR T cell only treatment or with an uninfected MSC control, or with my MSCs that were carrying the oncolytic and helper-dependent adenoviruses. I could look at a multitude of cytokines that were either effector cytokines, such as granzyme and perforin and interferon gamma, or also chemoattractive and regulatory cytokines, to where you could get an idea of what phenotype that your T cells were.
Results from the cytokine panel provided detailed information about the phenotypes of individual T cells in McKenna's samples. In samples pre-treated with the virus-containing MSCs, she found that the T cells had undergone unique changes. There were higher numbers of cells secreting effector cytokines boosting the immune response. In addition, a significant proportion of the T cells secreted more than one type of cytokine, which enhanced their activity. One especially intriguing result was the cytokine expression from CD4 positive T cells in the MSC-treated samples. Traditionally described as helper T cells, the CD4 cells surprisingly expressed effector cytokines at almost the same level as cytotoxic CD8 positive T cells, which suggested that the helper cells had a cytotoxic phenotype as well. McKenna next plans to improve the MSC Trojan horse to make the combination oncolytic adenoviral and CAR T-cell therapy efficacious in humans and across other solid tumor types.
The disadvantage of some MSCs when you deliver them to solid tumors is that they're quite large. And while we have utilized them to target lung tumors because their ability to travel there directly after intravenous infusion, these cells get stuck within the lungs and the capillaries. So what we would probably need to work is to modify the size of MSCs and then also some of the surface markers to prevent them from being so sticky once they reach that site. And then also, do we need to modify some of the stimulatory transgenes that we're providing in our virus? I think IL-12 and anti-PD-L1 have really hit a home run, but we may need to think about different types of transgenes as well. Future directions is to see how broadly applicable this therapy is going to be, not only to target lung cancers, but also to target other types of tumor. And not just utilizing our HER2 CAR, but also some of the other validated CARs that are being utilized in the clinic as well. I hope to really further expand on the spheroid data, in that I can really work to understand how oncolytic viruses spread within these structures, and then how that works with other different combination therapies.
Thank you for listening to The Scientist Lab Talk. This episode was produced by the Creative Services team for The Scientist and narrated by Niki Spahich. Thanks again to our sponsor, Isoplexis. To keep up to date with this podcast, follow The Scientist on Facebook and Twitter and subscribe to our podcast channel wherever you get your podcasts.